everyone, my name is Lucy Williams, and I will be presenting our paper, Flow Decomposition with Subpath Constraints. This is joint work with Alex Tomescu and my advisor, Brendan McGee. What we do in this paper is we initiate the formal study of the flow decomposition problem with subpath constraints. I'll first explain the basic algorithmic problem, and then I'll talk about its applications in biology, then go into our algorithms and experiments to validate it as a model for RNA assembly specifically. In the basic version of the flow decomposition problem, we're given a flow network as input, meaning that at each node, except the start node S and the end node T, we have conservation of flow. In this work, we restrict to DAGs and to positive integer flows. The task in the flow decomposition problem is to produce a set of S to T paths along with corresponding weights so that the input network is perfectly decomposed. In this example, we can decompose the flow into three paths, one with weight 9, one with weight 11, and one with weight 987. If we were to superimpose all three of these paths back into a single network, we would get exactly the flow network that we started with. And I'll also note that we use the variable k to denote the size of a ground truth set that created our input. We distinguish between versions of the problem where we simply need to find any decomposition and ones where we need to find the smallest possible. So for FD, we call that MFD. It's known that the decision version of MFD is strongly NP-complete because it's NP-complete even when the flow values come from only 1, 2, and 4. And it's also NP-hard to approximate, again, even when the flow values come from that same set. In this paper, we define the flow decomposition with subpath constraints problem, or FDSC. Here, we're given a set of simple paths, and we need to find a decomposition that not only decomposes the flow, but also covers all of the subpath constraints. In the example here, the pink path would be a subpath constraint. And finally, MFDSC is the minimum version of FDSC. So since MFD is just a special case of MFDSC, where we have no subpath constraints, MFDSC is certainly also strongly NP-complete. But while it's easy to see that FD can be solved in polynomial time, so just giving a decomposition of a flow network, not worrying about it being minimum, it's a bit tricky to decide even whether an FDSC instance is feasible, and we'll get more into that later in the talk. Now I'll explain the example application that we use to show the usefulness of flow decomposition with subpath constraints, which is RNA transcript assembly. Genes don't just code for a single protein, but instead they can sometimes create many different proteins through a process called alternative splicing. We focus on exon skipping, which is the primary mechanism for alternative splicing in humans, where the RNA transcripts are created by skipping parts of the protein coding portion of the gene. So for example, one gene could create 3 different RNA transcripts, like so, which would then be translated into 3 different proteins. Measuring the expression levels of these transcripts, or even predicting what transcripts there were, wouldn't be a problem if we could directly read the sequences, but in practice, we get reads, and so the transcript assembly problem is to predict the set of transcripts and their abundances directly from the reads. And additionally, in biology, there are some similar problems, such as viral quasi-species assembly and metagenomic assembly. So now we'll connect flow decomposition and RNA assembly. The basic model that's used in many tools to assemble these reads is called a splice graph, where nodes represent some subsequence and edges represent adjacency between the subsequences. Because we're just modeling exon skipping, our splice graphs are always going to be DAGs, and it makes sense to lay them out linearly like this. But in general, we could imagine that graphs used for metagenomic assembly or other problems could be cyclic. Now, by mapping reads that span two nodes, like so, to the edges that correspond to the junction between those two nodes, we can build ourselves what should be, in theory, a flow network. Then, decomposing that flow, for example, like this, maybe we would assign this blue path here a weight 11, this orange path a weight 3, and this green path a weight 2. So any decomposition would give us a possible set of transcripts in their abundances. Though, of course, what we're most interested in is the true set of transcripts. So it's generally assumed that the smallest path decomposition is the best. And in fact, this was validated in a paper that I'll talk about in a moment. Now, one issue with the splice graph model is that it contains many paths that are not the true paths. And so it's natural to think of using these reads, if they overlap 3 or more nodes, as subpath constraints, which would give us more information when we're trying to find the true set of transcripts. And in fact, some transcript assemblers are already doing this. Here's a list of some RNA assembly tools that use flow decomposition variations as part of their pipeline. And the two in the middle, Scallop and Transborrow, both use the notion of subpath constraint already. And I'll pause here just to emphasize that this flow decomposition step is just one part of an extensive pipeline for RNA transcript assembly that includes other steps that are challenging and important as well. So these tools are doing a lot. All right. One of the primary inspirations for this particular work is a 2018 publication where the authors also noticed that MFD was a common subroutine in transcript assembly techniques, but since the problem is NP-hard, it was only ever solved using heuristics, meaning that we didn't really know if the tools were doing well or poorly because MFD was a good or bad model, or for some other reason. So by giving an FPT algorithm and then engineering software based on that, which they called Toboggan, they could test that assumption that the MFD of the splice graph really corresponds to the ground truth solution. First, I'll give a high-level overview of their FPT algorithm. The idea is to enumerate possible path coverings and then check if there can be a weight assignment for those path coverings that perfectly decomposes the flow. So for a given path covering, we can think of having a linear constraint for every edge based on which paths cover the edge. For example, in the table on the right, the first row is saying that the blue path is the only path covering the top edge on the left between nodes 1 and 2, which has flow 1. And the second row is saying that the yellow and pink paths cover the bottom edge between nodes 1 and 2, which has flow 3. Each of these rows in the table on the right is an equality constraint, and the variables going along with the coefficients would be the weight assignments to each path. Because we assume that splice graphs are DAGs, the authors here can give a nice way to generate all of these path covers by topologically sorting the nodes and then using a dynamic program to extend paths as they go through. This figure is showing one way to extend from a path covering of the first 3 nodes to a path covering of the first 4. With an exact solver, they could test the parsimony assumption for FD in RNA assembly. And generally, they did find that MFD was a good model. Overall, in their synthetic dataset for human transcriptomes, only a tiny number had ground truth decompositions that used more paths than the optimal solution. But their software also finds the correct decomposition most of the time, and they also didn't report on instances where toboggan was unable to complete in a specified amount of time. So from their data, it wasn't possible to know exactly how many instances were not predicted correctly based on the decomposition being non-optimal, the ground truth being non-optimal, or based on there just being multiple optimal solutions and toboggan choosing the wrong one. So in our work, we dig a little deeper into their data and using their software, and we find that the errors when using MFD for transcript assembly are due to choosing the wrong optimal decomposition when multiple exist. This provides another reason to formalize the FDSC problem, as we do here, because just using flow decomposition, or MFD, isn't enough, as we have more and more transcripts, and so we can use subpath constraints to try to narrow down the true decomposition from amongst the optimal ones. So now we'll move on to our algorithms. So first, we just saw the usefulness of an exact solver to validate the MFD model. So in this paper, we extend the toboggan FBT algorithm to work for the MFDSC problem. So the idea here is that we can encode the subpath constraints as additional linear constraints and again solve integer linear programs or check to see if solutions exist to integer linear programs. And we call our implementation coaster. So to validate MFDSC for RNA transcript assembly, we simulate subpath constraints in the same data set that they used for toboggan, and we vary both the length of the subpath constraints, that's shown here, and also the number of subpath constraints. So when the number of subpath constraints is 0, MFDSC is just MFD. So these numbers in this column were also reported in the toboggan paper. And we see that accuracy goes down as k goes up, k being the size of the ground truth. But the addition of subpath constraints does indeed increase accuracy, as we would hope. So for example, when the size of the ground truth path decomposition is 7, as shown here, we go from getting only about 81% of the instances right when we have no subpath constraints, so in the MFD version, to getting 91% of the instances right when we do have subpath constraints with the And we have 4 of them, and they're the longer size. But coaster does get slow as k goes up, so we also developed a heuristic for the problem. The first thing we need to do here is define something that we call an over-demanded edge. So if the total number of subpath constraints covering an edge is greater than its flow, we say that the edge is over-demanded. For example, edge A to B here has 2 subpath constraints, but only a flow of 1. In this case, the over-demanded edge doesn't cause any problems because we can still decompose the flow with a single weight 1 path. But in this instance, we have 3 subpath constraints, the green one, the red one, and the blue one, and in order to satisfy them, we would need to use the entirety of those paths because they all go all the way from S to T. But the edge S to A, for example, only has flow 1, but we would be routing 2 paths on it. So this instance is infeasible. But according to our formulation of the problem, this would still be allowed. So a key to our approach to decide whether an input is feasible is this observation. If an instance is feasible, any over-demanded edge must be covered by 2 subpath constraints that are satisfied by the same path in some solution. This means that we can consider every possible way of merging subpath constraints on those over-demanded edges, and if we're able to do so without running into a situation where there are still over-demanded edges left, but no more merges available, we know we found a new instance where a path decomposition in that instance would also solve the original problem. And we can explore these merges recursively, which gives the runtime shown here. The last thing to address to get a sense for this heuristic algorithm is how we solve an instance that has no over-demanded edges, but does have subpath constraints. We do this by taking the instance and mapping it to an FD instance so that any solution to the FD instance can be mapped back to a solution to the FDSC instance. I guess this reduction here. And how we do this is for any subpath constraint, like this one in the example we saw earlier, we can just encode that as a new edge in a FD instance. So we can get rid of all of the subpath constraints and just solve an FD problem instead. Finally, we compared the accuracy of the heuristic with the accuracy of the FPT. This table shows some of our results. So the FPT accuracies that we already saw on a previous slide are the bottom rows, and the heuristic accuracies are the top rows. You'll notice that for a large K, like K equals 7, there is a big gap between the heuristic performance and the FPT performance, exact solver performance, but for smaller Ks, the heuristic actually performs quite close to the exact solver. To conclude, I'll tell you about some of our ongoing and future work concerning this problem. So the FDSC problem has us thinking about some other related problems that also have applications in biology, like using paired subpath constraints or subpath constraints that have demands that need to be satisfied. And in the future, we plan to apply our methods to more realistic transcriptome data. But that does present challenges because we need a perfect flow to start. So some tools already do this step as part of the process. So we could potentially adapt their methods, but we also have some new ideas as well. And if we want to test FDSC or MFDSC for other assembly problems, like viral quasi-species assembly or metagenomic assembly that we mentioned earlier, we're going to need to be able to decompose cycles, which does seem to be pretty difficult to do. And then finally, as we saw in our accuracy tables, as K gets large, even when we use an exact solver, we still don't get the answer all the time, the correct answer all the time, because we can't differentiate between multiple optimal solutions. So to increase accuracy, we're going to need to add more information using additional constraints or additional optimality criteria. And with that, thank you for listening, and I'll take any questions.